Okay, guys, the next slide is about the ketone bodies, which are a group of special molecules that can provide energy to the body under certain conditions. So first thing up, we need to know which molecules are called the ketone bodies. So there are three molecules, acetoacetate, beta hydroxybutyrate, and acetone, which is usually found in the gaseous state. So these three are called the ketone bodies, and they are synthesized in the liver and exported to extrahepatic tissues, means to anywhere else in the body except the liver. And it can be used as the energy source or as a fuel by other tissues, especially by the brain, under certain conditions. And those conditions are usually the conditions in which gluconeogenesis will be more active. In other words, conditions like diabetes mellitus or uh, prolonged starvation, these are the conditions where the body produces more than usual ketone bodies. So as I said, that ketone bodies are overproduced or produced more than usual during certain conditions, and these conditions are diabetes mellitus and starvation, when we are not eating for a long period of time. And uh, these conditions will cause a increase in the cycle gluconeogenesis. So you need to know a few cycles which we read in carbohydrate metabolism to understand this whole concept of ketone bodies a little bit better. So gluconeogenesis was a pathway in which we produced new glucose, and conditions like untreated diabetes and starvation are the conditions in which we have more gluconeogenesis than usual. So for gluconeogenesis, we use the molecule oxaloacetate, and when we are doing more gluconeogenesis means we will use this oxaloacetate extensively, more than usual. And when oxaloacetate becomes depleted, the acetyl-CoA will start to accumulate, because acetyl-CoA requires oxaloacetate to start the citric acid cycle. But liver only have a limited amounts of the coenzyme A. Therefore, what happens is, for citric acid cycle, we need acetyl-CoA plus we need oxaloacetate to start. But in these conditions, we do not have the oxaloacetate, so acetyl-CoA will be accumulated and it is not being used up. Therefore, one way the body uses this acetyl-CoA is to condense them together and make these ketone bodies, and therefore the coenzyme A will be freed as well, and later on can be used for fatty acid oxidation, so that will continue as well. Plus these ketone bodies that we just made can be used as fuel or energy in many tissues of the body, like skeletal muscle, like heart muscle, brain, and renal cortex. Brain can adapt to using these ketone bodies for energy under these emergency conditions. Next is the actual pathway by which we synthesize these ketone bodies in the liver, and this whole pathway is known as ketogenesis. So synthesis of ketone bodies in the liver is called ketogenesis, and we have got like a 3 or 4 reaction, a short pathway. First reaction, we join two acetyl-CoA molecules together to get acetoacetyl-CoA. Then we add up a third acetyl-CoA molecule to it, and it becomes HMG-CoA with the help of HMG-CoA synthase enzyme. Then this HMG-CoA will break down into the first of the ketone bodies, that is acetoacetate, which can be converted into the other two ketone bodies, that is the beta hydroxybutyrate and acetone. So this is a short pathway by which we get the three ketone bodies. One is this acetoacetate, then the beta hydroxybutyrate, and the third one acetone gas. So it's a simple pathway, basically using acetyl-CoA molecules to generate these acetone bodies, and happens in the liver, do remember, nowhere else in the body. So once the liver makes these ketone bodies, it will be exported to extrahepatic tissues, which can use these. These extrahepatic tissues are many organs, basically the muscles and the kidneys and especially the brain. So the beta hydroxybutyrate is the main ketone body that will give energy to these organs. So it goes or transported in the blood from the liver to other tissues which I just described, and in those tissues the enzyme dehydrogenase breaks it down into acetoacetate, and then acetoacetate is converted into acetoacetyl CoA. Now this beta ketoacyl CoA transferase enzyme is really important, and it will break acetoacetate to acetyl CoA, and eventually from here we get the two acetyl CoA molecules by thiolase. Now this enzyme is particularly important, the transferase enzyme, because the liver lacks this enzyme, and because the liver does not have this enzyme, therefore liver can only make ketone bodies, it cannot use ketone bodies, and this statement is written at the bottom here. Liver is therefore only a producer of ketone bodies, but it cannot consume it, or liver cannot use it for energy purposes. Only other organs can use it, and this whole breakdown process of the ketone bodies to get energy from them is known as ketolysis. So these ketone bodies are normally produced in the liver as well in every normal human being, but in a couple of conditions there is overproduction of ketone bodies. In other words, they are produced more than the normal amounts, and these two conditions are untreated diabetes mellitus, in which the patient's sugar levels are continuously high, and in starvation, means the person has not eaten for a long period of time. So when these ketone bodies are overproduced, they will be released into the bloodstream, or they go into the blood and increase the blood levels of ketone bodies. Out of the three ketone bodies, acetone is a volatile gas, and it imparts a characteristic odor or smell to the breath of the patient. So this could be useful in diagnosing diabetes, and in diagnosing this condition in which high ketone bodies are there in the body, and the high levels of the other two, that is acetoacetate and beta hydroxybutyrate, because both of these molecules are acidic in nature, they will lower the blood pH dangerously, and this condition is called acidosis. And these high ketone bodies in the blood and urine of the body will reach extraordinary levels. This condition is called ketosis, and joining these two conditions together, we get the ketoacidosis. So the acidosis in the body that is caused by the ketone bodies, and it can be very dangerous and can cause the death of the patient as well. So it can be fatal, and individuals on very low calorie diet, using the fat stored in adipose tissue as their major energy source, can also have high ketone bodies in their blood and urine as well. Next up is the regulation of the fatty acid synthesis and the breakdown. How do the body control these two pathways of breaking and making the fatty acids? So in the liver, the active fatty acid, or what we call the fatty acyl CoA, have two major pathways, or it can go in two ways basically. If the cell or the body wants energy from it, we can beta oxidize it in the mitochondria and get energy from it. And if it's extra and the body does not require energy, we can convert it into TAGs and phospholipids, and we can store it basically for later use. Now when you are eating regularly and the diet provides a ready source of carbohydrates, the beta oxidation of the fatty acids will be unnecessary, that is in regular diet which contains carbohydrate, and therefore this beta oxidation will be down regulated or be inactivated. So the regulation of the beta oxidation basically is through the step which is known as transfer of these fatty acids. The whole pathway is controlled by the rate of the transfer of the long chain fatty acids into the mitochondria, and this is what we call the rate limiting step. This three-step process or the transfer is known as the carnitine shuttle, which we discussed in detail. So in other words, if we want to activate beta oxidation, we will increase this transport from cytoplasm into the mitochondria. And if we want to stop this pathway, we will stop this transfer, because once inside the mitochondria, these fatty acyl CoAs or the fatty acids will be committed to the beta oxidation, means they will always be oxidized. So this carnitine shuttle or the transport process is the point of regulation. Basically, we activate or inactivate this transfer to control the whole pathway. Next is the short-term regulation of the beta oxidation, and there are mainly three molecules that inhibit this beta oxidation. So high malonyl CoA molecule will stop or inhibit beta oxidation by acting on this carnitine acyl transferase or the CAT1 enzyme. High NADH molecules, or we can also say high NADH/NAD ratio, this will inhibit the beta oxidation as well by causing the inactivation of this enzyme beta hydroxyacyl dehydrogenase, and high acetyl CoA as well. This will also inhibit one of the enzymes of beta oxidation, the last one, which was thiolase. Whereas in this slide, the only thing that will activate the beta oxidation is the low ATP and AMP level. So more low the ATP, it will activate the beta oxidation. In other words, when the energy levels are low in the cell. So the same four factors that we saw in the previous slide, high malonyl CoA, we can see here inactivating the CAT1 enzyme, which is going to transfer the fatty acid into the mitochondria. So this will stop the process. High NADH inhibits this dehydrogenase enzyme, and high acetyl CoA stops the thiolase, which is the last enzyme of beta oxidation. Whereas the only thing activating this whole process is the low ATP AMP ratio. Next is the long-term regulation of this beta oxidation, which is mainly done through the hormones. So this is the hormonal regulation, also known as the long-term regulation, and it will be triggered when the body has increased demand of energy from the fat catabolism. And this will happen when we are fasting in between the meals, or under long conditions of starvation. You have not eaten for a long period of time. So in such conditions, glucagon hormone will be released in response to the low blood glucose levels, and it is going to control this whole process of beta oxidation. So this whole hormonal regulation of the fatty acid synthesis and breakdown is done through two hormones, glucagon and insulin. And both of them work on the same enzyme, which is this one, ACC, acetyl-CoA carboxylase. And this enzyme does this reaction, converting the acetyl-CoA molecule into the malonyl-CoA molecule. Now, when the person has not eaten for a long period of time, or in other words, he's starving, he will have low blood glucose levels, and this will trigger the release of glucagon. And the glucagon's main effect is to inactivate this ACC enzyme. So when this enzyme inactivates it, the reaction will stop from acetyl-CoA to malonyl-CoA. So under the effect of glucagon, this reaction will not be happening. This acetyl-CoA will not be converted into malonyl-CoA. And when this reaction does not happen, the malonyl-CoA cannot stop this CAT1 enzyme, because when it is there, then only it will go over the mitochondria and stop the CAT1 enzyme. And when it cannot stop it, the fatty acyl CoA goes into the mitochondria and is beta oxidized, and we get more energy from these acetyl-CoA molecules. Now the opposite thing happens when we have high blood glucose levels. So when you have eaten a big meal, your blood glucose will be high, and we will have high insulin in such conditions. Now high insulin is going to activate this enzyme ACC. When we have activated enzyme, we will have more and more of acetyl-CoA into malonyl-CoA conversion. Malonyl-CoA goes there and stops CAT1 enzyme, and once it is stopped, this fatty acyl-CoA cannot enter the mitochondria. So its entry into mitochondria will be stopped, and whole of this pathway will be stopped. So the beta oxidation will be reduced basically under the effect of so this slide is basically have the text of the previous diagram which I just described it to you. So high blood glucose levels releases insulin. Insulin activates the ACC enzyme. This enzyme catalyzes the formation of malonyl-CoA. So we will have high levels, and this malonyl-CoA will inhibit the CAT1 enzyme, therefore prevents the fatty acid entry into mitochondria and stops the beta oxidation. The opposite effect is for glucagon. In between the meals, blood glucose levels will be low than normal. Glucagon will be released, and through the pKa, it will inactivate the ACC enzyme. When this ACC inactive, the malonyl-CoA concentration is going to fall, and it cannot stop the fatty acid entry into mitochondria. So fatty acids will enter mitochondria, and therefore beta oxidation will be increased.